Hi there, I'm Mrs. Irvin, and we're going to start off by me introducing you to a couple trees on my property. These are ash trees, and you can see they're the ones that are on either side of my driveway. When we bought this land and built our house, we had our driveway go right through two of them, and we nicknamed them the hello goodbye trees because when we're coming home, we say hello to them, and when we're leaving, we say goodbye. So these are our special trees. For 20 years, we've been saying hello goodbye to these trees. But recently, I found out that the ash trees in our area, and actually in many parts of the country, are in trouble because there's a bug called the emerald ash borer. Emerald because it's green, ash because it attacks ash trees, and borer because it bores in to the bark and gets into the tree and harms it and often eventually kills the tree. So I was very interested to find out that there are some local experts that work here at the University of New Hampshire right near us, and they are studying this beetle in order to understand more about it so that they can help to save the ash trees. So I thought this was very exciting to hear about, so I went out to meet them and to find out about their project. So I want to share that with you today. I'm going to introduce you to the team of experts, the scientists, who are actually, it's very exciting, they're working today out in the field trying to learn as much as they can about the emerald ash borer so that they can help save these trees, help save the ash trees.
Hello everybody, my name is Jeff Garnis, Dr. Jeff Garnis. I'm a professor of forest ecosystem health at the University of New Hampshire, and I'm here today to talk about some of the experiments we have going on in the state looking at the insects and the fungi that affect our trees. Here we're looking at a really important species, the species called ash. There's a few different species of ash, and then unfortunately there's a really nasty bug that's attacking our ash. And my team, students and postdocs and other people in our lab are working really hard to try to help to control the spread and the damage caused by the emerald ash borer, which is a tiny little insect, a beautiful green insect, but unfortunately a very damaging one. And we'll talk a little bit about how we're doing that in the videos to come. Okay, so here we are at the experimental site, or one of the experimental sites that we have here in Durham. This is Doe Farm. Many people probably know Doe Farm, right by the Lamprey River. This is Todd. He's a postdoc in University of New Hampshire, and he's doing a lot of the work and the heavy lifting with this project and asking some really cool scientific questions. So one of the motivating questions was, how do trees defend themselves, and do bigger trees defend themselves differently or more than smaller trees? So we have two different sizes of trees that we're studying here, which I think you can see. We noticed, as a pattern in nature, the small trees were not getting attacked by the emerald ash borer, this invasive insect, whereas the bigger trees, and even bigger than this, were very much being attacked and more quickly, and were dying more quickly, and were more heavily infested. And so we wanted to ask why. And one of the things that we thought was maybe the chemical defenses of the trees were different across the different size classes. So we developed an experiment to try to study that, and this is basically the last stage of the experiment.
Dr. Garnis explained in the video the pattern they observed in nature. The beetles were attacking the larger trees much more than the smaller trees. Somehow the smaller trees were able to be protected from the beetle. So this is very mysterious. Why were the larger trees dying more often? And so I want you to think about what you would do to design an experiment to make some discoveries about this question. How could you learn more? Why is this happening? So I'd like you to think about that and pause the video and think about how you might develop an experiment based on this question. And although their investigation is already happening, and they're kind of in the middle of it right now, they still could learn from you. So maybe you have some ideas, and we could send your ideas to them, and they might choose to investigate some of your ideas.
I'm going to pick this piece of tree up again. So this isn't the whole tree. This is just part of the tree that we cut down. And you probably noticed on this tree, it looks like there's little bands here. And what we did is part of our experiment to study how different sized trees might be able to survive an invasive beetle that wants to feed on them is we actually put the beetles on the trees. And the reason we did that is that allows us to know exactly how many beetles are in the tree, as well as control different aspects of our experiment. And so we put these beetles on the tree because we wanted to see how, when a beetle attacks the tree, it affects its chemistry, like Dr. Garnis mentioned. And so when trees are attacked, they have chemicals in them, just like we have an immune system that helps protect them against things that might not be good for them, such as a beetle that wants to feed on them. And so we put them on the tree here. And the reason these bands are here is because we actually wanted to give the beetles a little bit of a leg up. We wanted it to be easy for them to get into the tree. And sometimes bark is pretty thick. And if you're an insect trying to lay your eggs on the bark, and then you have to dig all the way into the tree, which is where these beetles feed. They feed under the bark inside the tree. We wanted to make it easier. And by doing that, we would hopefully have more beetles survive and get into the tree for our experiment. Now what we're doing is we're actually peeling the bark back. So we have special tools that we use that allow us to take the bark off very carefully and safely. And when we do that, we can uncover what's under the bark. And there's a type of tissue under there that the beetles feed in. It's their food. They also live in it. It's called phloem. And we can look at the phloem tissue under the bark of this tree, and we can study the beetle. And so there's two things that we're really interested in with this beetle. One is the tunnels that they create, which we call galleries. And we're looking to see how long they are. If the insect's feeding for a shorter distance or a longer distance, that could mean that they consume more or less food, which may make them a healthier or less healthy adult beetle when they come out of this tree, or a standing tree that was not cut down next year in May or June. And then the other thing that we're looking at is to see how big and how developed the beetles are. So just like humans, insects have different developmental stages that they go through. And enough about these beetles that we can tell, which stage they're at. And so we're looking to see, for instance, in a big tree, like the one I'm holding, do the beetles develop faster, or is it a more slow development? And that might be because of a tree's ability to defend itself against the beetle. Another part of this project, which hopefully we'll get the opportunity to show you, is there are wasps that have been released to help control this beetle. They're not the kind of wasp that you would probably be used to, such as like a yellow jacket or something that you may have been stunned by, or somebody you know. These are very, very tiny wasps. The biggest one would fit on my pinky nail, so they're pretty small. And even some of the smallest ones look like about the size of a grain of rice. So if you were looking for them and you didn't know they were there, you might never see them. But as part of this project, we release these wasps because we're interested in seeing how effective the wasps are at controlling this beetle and attacking it to protect the trees. And we're interested if they have a better, if they're better at it in larger trees, or if they're better at it in smaller trees. And so how well a tree can defend itself can also influence how successful the wasps that eat the emerald ash borer, the invasive beetle, and that's something that we don't really know. So that's part of this project that we're studying. And so to actually tell you what we're doing right now and show you, in addition to these bands here, I mentioned we're peeling this bark. Before we do that, every one of these logs, which is pretty big, this one's about 3 feet long, is measured so we know how long it is, and then we measure the diameter to see how wide it is. And that allows us to understand exactly how much of the tree these beetles have fed on. And then another thing that we have been doing, which I will show you right here, I am using a pretty standard tool that a lot of engineers or a lot of other people that measure things use. It's called a caliper. I'm going to hold it a little bit close here. It opens and closes so we can get how big things are. We can figure out But the thing that's nice about it is at the bottom here is we have a little piece that comes out. And so that allows us to measure how deep things are. And what we're doing is we are taking the bark here, and each one of these little ridges along the log, we are measuring how many of them there are and how deep they are. And what that'll allow us to do is get a sense of how rough or smooth the bark of a tree is. And how rough or how smooth the bark of these trees are might also be a thing that influences how successful the beetles that are feeding in here are. That could protect them on thick bark from getting attacked by the wasps that we released, or on a smooth tree that might make them able to be eaten.
All right, so now we've heard the questions that these scientists have, and they keep developing more and more questions as they learn more and more. But we've also learned about this method they're going through, these series of steps that involve tools and observations and keeping records, so that how they can learn as much as they can about what's happening with the beetle and how we might go about helping to stop the beetle from harming the ash tree. So as we continue watching the video, I want you to listen for the things that they're doing, the tools that they're using, anything you notice about what scientists do to learn more about what they're wondering about. And maybe you might have a question someday, and you might set up your own investigation or something with a classmate. You might do it in school, you might do it at home, talking to your parents about something. If there's something that you're really wondering about, you could also start to think about how you might go about learning more.
Here we have a log that has already been measured. We know how long it is, we know the diameter of each side of the log, and how rigid it is. And this is Casey here. Hello. Casey's another scientist in our lab working on this project with me and Dr. Garnis.
What I've done is I've uncovered the gallery, the feeding tunnel from the EAB, and I've peeled it back very carefully, and I've exposed this tiny larvae right here. And I don't know if you can see it from there, probably not. But what I would do is I would extract the beetle, and we would save it to later measure it and kind of compare with the other beetles that we find. But likely this log right here has many of these feeding galleries, and my job is to uncover all of them and measure them and to complete the study.
So that was made by that canal. That's what you call it, a canal? Did you say that?
It's a gallery. It's
gallery.
typically serpentined, like snake shaped, but that's kind of the signature shape of the feeding gallery.
And so the boars go in there and they eat the phloem.
Yes.
And they travel and they make that path. And you can't. It's hard to see if you're just hiking in the woods.
Yeah.
So it's more you have to really peel off the bark to be able to.
Yeah, and what you
see
can
it.
notice is woodpeckers will actually go after these, and if you look at some ash trees that have likely the beetle underneath, you'll see some of the bark being flecked off by the woodpeckers, and it's kind of a sign that EAB is there and they're being fed upon.
Okay.
Yeah.
You might want to think about why we would want to save or to help a certain species of trees. We've got lots of trees all over. We've got oak and maple. There's a lot of pine and hemlock in New Hampshire. There aren't a whole lot of ash trees. Actually, they make up about 7% of the trees in New Hampshire. But one reason you might want to think about why you think it's a good idea to have many different kinds of trees, but often it can help to make our forests safer. There's something called biodiversity when there's kind of different kinds of trees. It can bring different kinds of animals, different insects. It helps to make our forests healthier. And as you know, trees in general take in our carbon dioxide in the air, and they release oxygen for us to breathe. They help with all sorts of things, with keeping our soils healthy, with helping with the water table. So they're very important for us as humans as well, to have these trees in our lives. So First, I want to take you up close to this tree. Right now, you cannot see the leaves because the leaves have fallen off, but the leaves are very interesting. They look quite different than a maple or an oak. They have leaflets going off that grow opposite to each other on the branch, so that's one way to kind of identify them, is to look for branches and leaves that grow opposite to each other. The other thing is they've got this really cool diamond-shaped bark that I'd like to show you. And here's a close-up look of the tree. Some people would describe this pattern of the bark as having diamond shapes, that there's these diamonds kind of scattered all along this tree. This tree was measured the other day. I took a measuring tape, and it was 116 inches all the way around it. So its circumference, or the distance around the tree. I'm really interested now in getting to know these trees more since I've found out about this study.
Okay, so I'm actually doing some of the hard labor here, which I sometimes don't get a chance to do, but I'm peeling a log, an ash log that has been infested with the emerald ash borer, intentionally so that we could study how it affects the trees and how it interacts with the trees and the trees' chemical defenses, as you just heard. So what I have here is a draw knife, a very sharp tool with two handles that allows me to pull towards myself and take off the outer layer of bark, including the phloem. So the phloem, it has lots of sugars. This is a good place for insects to make their living and feed on. If you peel right down to the face of the wood, that's the xylem layer. And the emerald ash borer lives between the bark and the xylem layer. So in this very thin area, that's their food resource, the phloem. That's where they live, and that's where they spend their entire larval life cycle. And then just at the last stage, they actually dig into the xylem a little bit. And part of the reason that they do that is to get away from their own natural enemies, so that it's harder for the woodpeckers to reach them, harder for the parasitoids to reach them. Honestly, the woodpeckers don't mind that much. They just dig as deep as they need to. But the parasitoids, the wasps that infest these guys or infect them, may have a bit harder of go of it to try to get through the xylem and find the larvae. So we're pulling off the phloem and very carefully looking for the signs of the emerald ash borer, the galleries, the serpentine galleries that we find. I'm not finding any here. And then we also have to be very careful to even dig down into the xylem layer, into the wood layer, because that's where they pupate before they become adult beetles. So this is a special tool. This was specifically made for this purpose. It's a peeling table or a peeling chair, bench maybe, that allows me to secure the log tightly by pushing my foot down on this pedal here, squeezes the log in between these two pieces so that I can have a good hold on it, and it's much easier to peel. And these are quite sharp instruments. You don't want to get your finger stuck in there, but this is a little bit of a delicate operation because we want to be able to measure all the larvae as we collect them. So I have to be careful that I don't just chop their little heads off as I'm going, but you get pretty good at knowing when you're getting close to a larvae. You can tell little changes in the dryness of the phloem, and you can find the galleries quite effectively. So it's careful work, it's slow work, but it's actually pretty fun to be out here doing this. So,
hello again. This is a log that we completely scraped all the bark off of. And so you can see it looks completely different now. They actually have a different sound too. But on this log, there's a whole bunch of galleries that we were discussing here. And so I'm going to hold the log and get real close and show you. So as you can see, there's a whole bunch of different galleries here. So that means that each of these things is actually a different beetle that fed here. So we have a whole bunch that were in this tree. And we were just talking about as the beetles get more developed and right before they become adults, they have a period of what we refer to as pupation. A lot of insects, when they develop, they look completely different when they're younger to their adult form. So when we find the emerald ash borer in these trees, they kind of look worm-like or like a little maggot. But when they come out of the tree, they're actually really pretty. They're an emerald color, which is why we call them the emerald ash borer. But in here, right before they pupate and become adults, we can actually tell. It's really cool. So in the gallery here, which is again the tunnels that they make as they're feeding, you can tell when they fed and where they fed. And you can tell that because the older parts, so when they were younger, it's darkened. So this is what we call frass. That's their waste. They just pack it behind them. And when it's darker, that usually means that that's where they were a longer period of time ago. And when it's lighter, that's actually the most recent place they fed. And the one way that we can tell where exactly these beetles are is this light frass. So what we're going to do later is actually take a chisel, which is another tool that we use to work with the logs. It's dangerous, so we all work together in case anybody gets hurt, that they can get help if needed. But we actually chisel this wood out, and the beetle is under it, and we can pull it out. And this is exactly how we know where it is. And maybe I can show you where exactly that is in a second. But I just wanted to also point out that some of these beetles make really smaller galleries, like this is about this long. Whereas we see this one here it goes from about here to all the way up here. So we don't really understand or know why the beetles decide to feed across a larger or a smaller distance. But we know they do. And that's one of the things that I'm interested in, is I'm interested in the behavior of these insects when they're under the bark feeding. And what we do with these galleries, in addition to pulling the beetles out, is we take a whole bunch of pictures. And then we bring them on the computer, and we just trace this. And we can then measure exactly how long it is. And we're trying to figure out if longer galleries have some sort of positive benefit for the beetle, or perhaps tree defenses stunted their growth. And the food that they were eating was actually not very nutritious, and so they had to compensate. It's like you eat something that's just, it might taste good, but it doesn't have a lot of calories. And so they might be compensating by eating more to still reach the same developmental stage. But that's something that we hope to find out here, and that's something that we hope will be interesting to the scientific community once we complete our project. So again, this is a log that we are studying the galleries in. These two marks that you may have noticed are where we sampled the chemistry of the tree at 2 separate times this summer to better understand how the tree is defending itself. But right now, that's just sort of an artifact. It's just there, but you can see it, so I definitely wanted to point it out in case you had any questions. Okay, everyone, here I'm on the bark peeling station that Dr. Garnis was just at, and I am going to show you about some of this light-colored frass and show you what's underneath it. So again, I'm using a mallet and a chisel, which I said, can be dangerous. So if you use something like this, make sure you do it a safe way and with an adult. So what we'll do here is I know that the insect is right here, so I'm going to go a little bit ahead of it here. And then I just sort of cut out almost like a little rectangle. So I'm going to do a little bit here. And a little bit here. And so I'm just slowly trying to carefully cut this out so I don't damage the insect underneath. So the insect under here is, it's what we call a jay larva. So it hasn't quite pupated yet, but it's getting close to it. And they turn their body into a jay. They just sort of wrap themselves up here. So now I'm going to sort of peel this back. And, it takes a little trial and error. You got to keep poking at it. Let's see here. So I haven't found it yet, but we just keep hoping and keep carefully chiseling away here.
Right here.
And so I haven't found it yet. And so what I will do is I'll just cut this piece off. And then I take it and we see this little bit right here. That's generally the beetles dig a hole to help themselves get out before they leave. So we're going to carefully split this piece of bark, or a piece of phloem rather, in two and see if we can find it. Sometimes it's a little bit of a, you got to figure it out, just go slow. They're not always in exactly the same place that you expect them to be. So you got to be a little flexible. I think he's right in here. Let me just keep splitting this up.
Is that the stage they are in the winter, the jay larva, or?
Usually, but not always. Okay. It depends on a whole bunch of things with the environment. So, you guys, I know the light's changing a little bit, but if you see right there, there he is. He's in the side.
Oh,
I'm going to pop him out. So sometimes they're really deep in the wood, and you do that, and you take them right out. Other times you have to carefully open it up to find them. And here we are. So I'm just going to open this up for you here. We'll just keep opening up. And what we have, I have a special type of grabber for the insect. They're called featherweight forceps. Oh, thank you. And they're kind of like tweezers, but they're soft. So now I can pull the insect out. And it's right here. So it looks like it might have got chopped up a little bit. That's only half of it. The other half I think is still in there. So I'll see if I can hold it a little bit more. So this is the head right here. You can see it has the little dark part of the mandibles.
Is that like the teeth? Kind of.
Yeah, that's what they use to
insect?
chew through the wood.
Okay.
So that's part of him. So this is what we were saying. Sometimes they unfortunately get squished as we're pulling them out. We don't need them for anything specific. We are interested in doing some, maybe weighing them in the lab after. So I want to make sure I remove the whole insect.
Do you put them into a bottle or something and then take them
Yeah,
back?
we have little bottles with ethanol. Yeah, so I hope you could see that. I know they're kind of creamy white and it's bright out here, so it's kind of hard to see. But that's how we use our knowledge of their behavior and their biology to find these insects so we can remove them from these trees and study them.